Hello everyone, I Dr. Shamishta Banerjee welcome you all on the SWAYAM course on genetic engineering and its recent advancement. Today there is the lecture 1 of this particular course. Here I will discuss what is introduction to genetic engineering and the subtopics involved in this are what are genetic engineering, what are GMOs, the steps involved in its creation in brief, the genetic codons or code to give you an idea what are they and how they will be helpful for genetic engineering, the applications of genetic engineering and the examples of GMOs in different fields, how they can be fruitful to us. Then it is the process by which the pieces of DNA are transferred from one organism to another. Like it can be transferred from human to bacteria, bacteria to plants, human to animals like sheep and any other animals, from even fishes to plants, from even fishes to higher mammals or higher animals, it can be any. Just to give you an idea, here is a pictorial representation of the same. The definition of genetic engineering is, it is basically the direct modification of an organism's genome. Now the question is, why we modify an organism's genome or what is genome? Genome is the sum total of the genetic constituent which is present in an organism, or I can say it is the sum total of the genetic organism which is present in a cell. And why I am saying either it is a cell or an organism, because every cell is identical in an organism if we talk in respect of genome. So, basically genetic engineering is modifying this particular genome. So, why this genome is modified? That is, it reflects the specific traits or characteristics like eye color, drought resistance, skin color and many more. These are basically the traits or characteristics and which are basically reflected by our genome through the genes that are stored in the form of DNA. What are genes? To all of you basically know what are genes, but to again give you a quick glimpse of it, genes are basically the functional unit of DNA. So, genetic engineering is basically modifying the genes to transfer a particular characteristics which is not present by default in that particular organism. Changing the genome can enable engineers to give desirable properties to different organisms. Organisms created by genetic engineering are called as genetically modified organisms, or in short it is abbreviated as GMOs. Then the next thing I am going to discuss is the history involved in the development of these genetically modified organisms. These are the landmark discoveries of the field. In the 1973, the first time the genetically modified bacteria was created. In 1974, genetically modified mice was created. In 1982, it was the first time commercial development of genetically modified organisms started. Like we can say it was a land changing revolution in the field, the production of insulin in bacteria. We all know that during the past time the insulin was of very high demand because of the treatment of diabetes and it cannot be fulfilled naturally. Hence, there was a need for certain solution and which was solved with the help of these GMOs. In 1994, the first time the genetically modified food started to be sold in market. In 2003, again it was the first time when these genetically modified organisms as pet was commercialized. Like we all know, in an aquarium the glowfish which we put, they are basically an example of these GMOs itself. Now, what is the process which is involved in the creation of these GMOs, I will be discussing today in short, but in later on modules we will be discussing these steps in details. So, what are GMOs? All genetic changes affects the protein synthesis of an organisms, and if the protein synthesis is affected, the traits which they carry or the desired characteristics which will be present in an organism can be varied. So, we are targeting the protein synthesis process by modulating the genes for the creation of GMOs. So, by changing which proteins are produced, genetic engineers can affect the overall trait of organisms. Genetic modification can be completed by a number of different methods, which are as follows. Either a new genetic material can be randomly inserted, or it can be very specific or targeted location. Their second method involves the direct replacement of genes, like if there is a non-functional gene in an organism, we can directly replace it with the functional gene from some other organism, and the process behind it is basically recombination, or we can call it as homologous recombination. I am not going into detail of that particular thing right now. The third technique is removal of genes. Then the last one is mutation of the existing genes for beneficial purpose. Then there is a diagram which is being showed here, what is contained in a cell. There is a nucleus which is the master organelle of a cell, and this nucleus contains chromosome, and these chromosomes contains the functional unit in the form of DNA, and where the genes are present, and it is actually modified for GMO creation. Here in this figure we can see the code. What is this code? This code is very important, or it is very unique, we can say. We can see the 3 letters in this particular figure, and these 3 letters hold significance to us. What are these 3 letters? It depicts the amino acids, it depicts the 20 amino acids which are present in an organisms, like AUG, UAA, UAG. We can see the combination of 3 letters. All these 3 letter triplet codons are basically formed with the help of nucleotide bases, or we can say there are 4 nucleotide bases: adenine, guanine, thymine, cytosine and uracil. So, adenine stands, A stands for adenine, T stands for thymine, G stands for guanine, C stands for cytosine and U stands for uracil. So, these 64 codons are basically the combination of these letters only, but they are having a great meaning. We can see from this table, from this figure, that is every 3 letter codon codes for a different amino acid, like LEU, PHE. LEU stands for leucine, PHE stands for phenylalanine and so on. These are basically the amino acids. So, these code will depict what type of protein an organism can synthesize. All organisms basically used this same code book itself. Their genes will be read in the same way itself. And if we talk about human genome, with the combination of these letters, the DNA is 3.2 billion bases long, and these 3.2 billion bases long DNA is compressed in the form of 24 chromosomes. Now, the question is, can we mix the genes from one creature to another? After learning all this, the answer is certainly yes. Why mix genes together? Genes produce proteins in different organisms or different individuals, as in I have already discussed the example of humulene. So, we can see in this particular figure that initially the humulene or human insulin gene was not present in bacteria, but the human insulin from bacteria can be developed by transferring this gene into the bacteria. by isolating this particular insulin producing gene from humans into bacteria, and as a result human insulin or humulin can be produced, or we can say the genes are basically mixed here for medicinal purposes. Other example of this particular gene mixing is the bacteria which is capable of producing human growth hormone or somatotropin. I am going to discuss what are the uses of genetic engineering. It enables plant to produce new proteins, protects crops from insects like BT corn was produced. This corn produces a bacterial toxin that kills the corn borer, caterpillar pest of corn. Extended growing season crops can be created. Example are fish berries. So, what are these? Strawberries with an anti-freezing gene from flounder was created. The food quality can be improved. Example is golden rice. In this rice, the vitamin A was enhanced and its nutritional value was improved. The genetic engineering technique which can be used for the production of these GMOs are as follows. There are basically 4 methods. The first one is artificial selection, second one is cloning, third one is gene splicing, and the last one is gel electrophoresis, which is not a genetic engineering technique, but it can be used for analyzing the DNA. So, the first method is artificial selection. From the name itself, what does it mean? That is, herein we are artificially selecting a particular gene for breeding, and there are 3 subtypes of this particular technique. The first one is selective breeding, second one is hybridization, and the third one is inbreeding. The second technique is cloning, where the identical copies of an organism is created. The third technique is gene splicing. From the name itself, a particular gene will be spliced off, will be removed. And the last technique is gel electrophoresis, wherein the DNA can be analyzed by their charge to mass ratio under the influence of an electric field. It is a separation technique. So, with this we can identify whether the new DNA has been inserted into the host or not with the help of running the standard DNA ladders. The first technique is selective breeding. From the name itself, here the selected organisms are bred with one another. The second technique is hybridization. Herein, 2 individuals with unlike characteristics are crossed to produce the best in both organisms. Example, Luther Burbank created a disease resistant potato which was called as the Burbank potato. He crossed a disease resistant plant with one that had a large food producing capacity. So, what was the result which was obtained is the disease resistant plant was developed that can make a lot of potatoes. Other examples of hybridization can be the development of liger. From the name itself, what does it mean? Lion and mixture of tiger. Then the second example of this hybridization can be grapple, that is, it is again a combination of grape and apple. This fruit will taste like grape and it will look like apple. The third technique of artificial selection was inbreeding. Breeding of organisms that are genetically similar to one another was inbred deliberately to maintain the desired characteristics or traits. Dog breeds are kept pure in this way. It is how a Doberman will remain a Doberman. It keeps each bred unique from others. But the risk associated with this particular technique can be, since both have the same gene, there is a chance that the baby which they will develop will get a recessive genetic disorder, and the probability of occurrence of this disorder will be very high. The risks involved in this particular disease is blindness and joint deformities. Then I had already told the cloning definition, but I will like to add on here, that is identical twins are naturally created clones. They will have the same exact DNA as that of the parent. Then what are genetically modified organisms, or we can say what are genetically modified bacteria? Bacteria are most common GMOs because of their simple structure and it permits the easy manipulation of their DNA. One of the most interesting uses for genetically modified bacteria is the production of hydrocarbons like plastics and fuels, which are usually only found in fossil fuels. So, to make their demand sufficient in the market, these hydrocarbons needs to be produced in large quantities, and that can be possible with the help of these GM bacteria. And the example of these GM bacteria which was created was cyanobacteria, or blue green algae were modified to produce plastics, or more specifically I can say polyethylene was created, and the fuel production was also enhanced by cyanobacteria, and the fuel which was produced in this way was butanol, and they were produced in the form of byproducts of photosynthesis. Then the next example of GM bacteria is Escherichia coli have been modified to produce diesel fuel. The examples of GM crops which have already been developed are insect resistant, herbicide resistant, drought or freeze resistant, disease resistant, higher yield, faster growth producing GM crops, improved nutrition, longer shelf life. Insect resistance, I had already told the example of it. Herbicide resistance, from the name itself they will be tolerant to the herbicides. Drought or freeze resistant, they are basically needed or they are basically required in certain areas which is drought prone, so that the crop can survive in that particular condition. Disease resistant, we all know that majority of the agriculture crops are destroyed by many diseases or insects or pests. So, this GM crops if developed can be very beneficial for increasing the yield, for faster growth I had already discussed, for improved nutrition it has been discussed in the previous slide. Longer shelf life I will cover here, that is the flavor savor tomato were basically created for longer shelf life. We all know that tomatoes are perishable, they are destroyed very easily, and in shortage of the storage condition these tomatoes, their prices they go higher and higher. So, a flavor saver tomato was created where a delayed ripening process involving gene was inserted to enhance the shelf life of tomato. GM animals, the examples of this GM animal can be the development of bioluminescent animals. So, a luminescent protein containing gene was inserted into a mice, and the result was the mice which was created was giving the luminescence phenomena. So, what are the uses? It can be used for protein tracking, it can be used for disease detection using bioluminescent imaging to identify the different types of cells, that is to distinguish the diseased as well as the healthy cells, as well as the glowfish can also be created in this way, we had already discussed. Other examples of GM animals are the fast growing salmons. How they were created? Genes from two other fishes were inserted that caused this salmon to continually produce growth hormones, so that their size increases, and it was monitored or compared with the help of continuous measurement of their height and length and weight. How less smelly cows can be created? We all know if we pass around the cattle area, it smells badly, and it is because of a methane producing bacteria. So, with the help of genetic engineering, these bacteria can be modified for less production of methane, and as a result less smelly cows can be created. Now, there is a more fascinating example, that is, could Spiderman be real? Yes, to a certain extent we can transfer a gene from spider to a human, but transferring a single gene cannot lead to the production of a Spider-Man. There is another example of web producing goats, or we can see that biosteel are basically created this way. What does it mean? The fibers which are present in web, they are basically spun in the goat milk to create biosteel. Then here is a pictorial representation of what is genetic engineering, what are its applications, what are the steps involved in it, and we will discuss each of them in detail later on. So, the examples which are involved here, we can see that an anti-clotting protein can be created, or we can say that the growth hormone can be produced in large amount, or we can say that insect resistant or pest resistant plants can be created. Now, the most possible hazard or the most possible risk of the genetic engineering is there are some concerns related to it. It can be a risk for human health, that is, it can lead to have higher chances for cancer occurrence. The question is how? Basically, we are integrating the genes from some other organism, and if this integration is not at proper site, then it may lead to the stimulation of some genes which is not needed in natural case, and in this way it can even lead to the development of cancer. There can be risk for human health, like higher chances of infection can be there. There can be risk of creating new microbes that are difficult to kill by antibiotics, or we can say antibiotic resistant microorganisms may be developed because of genetic engineering. So, these are the very risky concerns. That is why there are certain ethical concerns, or in some countries these GMOs are banned. There can be a risk to human health as it is unsafe for intake. Nearly 50 countries around the world, including Australia, Japan and all of the countries in the European Union, have enacted significant restrictions or full bans on the production and sale of genetically modified organism, food products, and 64 countries now have GMO labeling requirements. It is because of these concerns. It can be harmful to environmental and wildlife, like it can create some new variety of organisms which are not safe. Increased pesticides and herbicide use can be forced because of this GMO development. Farmers' health is at risk, seed and pollen drift will be there, creation of herbicide resistant superweeds. What about genetic engineering in humans? And I am leaving this question for your thought process. Let's have a quick recap of what we have studied till now. Genetic engineering is a process that uses laboratory based techniques to alter the DNA makeup of an organism. This may involve changing a single base pair, that is adenine, thymine, guanine or cytosine, or deleting a region of DNA or adding a new segment of DNA. It is basically used in research and industry. It has been applied to the production of cancer therapies, brewing yeast, genetically modified plants, livestock and much more. Now, I would like to share some interesting facts about genetic engineering. Genetically engineered things are actually all around us. These techniques are widely used today for research, agriculture, industrial biotechnology and medicine. For example, genetically modified enzymes are used in laundry, detergents and many other fields. Some of the most common animal test subjects for genetic engineering are mice and zebrafish. Both have relatively short lifespans, and so any modifications to their DNA can be accessed very quickly in the growing animal. Zebrafish are particularly useful as their larval phase is more or less completely translucent. This allows researchers to easily see what is going on inside the infant fish as a result of genetically modifications which are made. Genetic modification is something of an ethical dilemma. While the practical benefits of genetic engineering are readily demonstrable, there are some other ethical and ecological concerns around them. For example, genetical modification of human embryo is rightfully considered unethical. With regards to ecology, it has been argued that genetically modified organisms, if ever released into nature or environment, could readily outcompete the wild native organisms. This could be devastating for natural habitats as well as species. Genetical modification researchers can build completely synthetic genomes. Genetic engineering has come a long way since 1970s. Today, researchers can actually build a long base which is cheap and accurate on a large scale. This allows genetically modified researchers to be able to conduct experiments on genomes that do not actually exist in nature. It is called as synthetic genomics. This field of study is really finding very interesting and getting much more attention nowadays. Some companies like Synthetic Genomes have been founded to study potential commercialization opportunities from custom designed genome. Genetically modified scientists can even build complete chromosomes in the lab. Further to the above, genetic engineers can today create entirely synthetic chromosomes in the lab. For example, the first synthetic chromosome for yeast was created only a few years ago. This is considered as a very big deal in the field of synthetic biology. It can be used on any kind of organism. Genetically modified animals are helping with some very serious human diseases and disorders. It is very sophisticated form of cut and paste technique. This we will discuss later on. What do I mean by this sophisticated form of cut and paste technique? This is the team of our instructors. We will be covering the different topics of this course one by one. The different instructors are Dr. Tanushri Chatterjee, who is the coordinator of the course, myself, the co-coordinator of the course, Dr. Pratima Gupta, Dr. Neha Sharma, Dr. Akanksha Vani, Dr. Ashutosh Kumar, Dr. R. G. Brajesh and Ms. Shilpi Banerjee. Thank you for your patience hearing.